Ahí está. Good morning, everybody. Welcome to this new edition of Sembrando Ideas. This time we have to do it in English, so I'm sorry of my Spanglish, but I will try to do my best. And that's because we have with us a special guest from Florida Atlantic University, Dr. McCoy, who I will be introducing to you in a few moments. The objective of today's meeting is to learn about and discuss how our panelists have contributed to SDG 3, good health and well-being, through their research work. After this brief welcome, let me, I have a technological problem, please. Okay, sorry. After this brief welcome, I will be introducing each of our panelists, and they will make their presentations. After we have all of our three panelists have made their presentations, we have probably the richest part of our meeting. That's the space of questions and answers that we have with our audience. And we will have like 20 minutes for this purpose. And after that, we will have our closing final remarks. So I want to give you some brief instructions before starting our meeting. Please keep your microphones off. That will help us during all the meeting. Please also, you are welcome to submit your questions for any of our three panelists at any moment during the meeting, but please submit them using the chat of Zoom. Our panelists will answer those questions during our question and answer space after all the presentations have been made. And finally, it's important to remember that the opinions expressed during this meeting are the exclusive responsibility of those persons who are expressing them and do not necessarily represent the thinking of ESPOL. Once say that, we will introduce our first panelist, Dr. Mercy Barbour. She is an oceanographer with a PhD in environmental science from State University of New York. She's a professor at the Escuela Superior Politécnica del Litoral, where she also is a senior researcher at the Pacific International Center for Disaster Risk Reduction. She has been a public officer, and because of that, she has been involved in decision-making processes at the local, regional, and national level in the areas of environmental management and climate change. She also served as the Chief of Environmental Control of Guayaquil, and she was the Provincial Director of Disaster Risk Management for Guayas, the province of Guayas in Ecuador, and also the Vice Minister of the Ministry of the Environment of Ecuador. Her research is transdisciplinary with a focus on the social ecological systems, space-time analysis for her studies of climate variability and vulnerability, coastal urban systems with an emphasis on the interaction of climate and human health and climate risk reduction. She's the lead author for the Assessment Report 6 in the Mitigation Working Group IPCC, and she is a member of the Human Resources Group for Climate Services of the World Meteorological Organization. So let's welcome Dr. Mercy Barbour. Mercy, your mic is off.
Okay, sorry. Okay, so thank you, everybody. Thank you, Carlo, for your nice introduction. And I'm very happy to be in this panel to discuss issues related with health. So I'm going to directly go to my presentation, no? And let me put it in big presentation, and let's go to discuss a little bit. I will try to discuss related how the research that we are doing at the center of the Pacific International Center for Disaster Risk Reduction is linked with human health, but also with climate change. So our idea is to present a framework that will be very useful to do in the policy arena, but also in the research arena, to integrate, these issues of climate, disaster risk reduction, and sustainable development goals. So let's define what is, say, the SDG 3. Good health and well-being is ensure healthy life and promote well-being for all at all age. And that's what they're looking at is to achieve universal health coverage and access to quality health care. No one must be left behind. It's good to see this because that's our goal as a global health, society. But the truth is, still we have a lot in debt. We have 431 million people in the Americas region that still do not have any access to safe sanitation. We have 161 million people that still do not have access to safe drinking water. The result of that is the 30,000 preventable deaths we have each year. So this is the issues that are behind of the human health, what is still there is a missing, addressing the missing of social issues that are in all the countries in the America. But in addition to that, 2020 was a full year fully of disaster. So we have drought in Mexico, hurricane in Central America. We, move around all the Greek alphabet, and we have a lot of impact in the coastal and also in the island. We have floodings everywhere in Ecuador. We have heat wave in Bolivia. We have a lot of fires, wildfires in Brazil. And the total disasters in South America count millions of dollars, but also 100,000 people. So we are looking at our thermometer that we're increasing year by year, and the heat is hitting us and all the people, especially to the most vulnerable. So in this context, I would like to go in this pathway of development, looking at this relation that sometimes is not very evident between this climate risk, this disaster risk reduction, and human health. And in this diagram that is from the working group 2, the recent assessment report number 6, we have that the risk of these climate issues are related with the hazard, of course, the heat, the flows, the storm, the vector spread, but also they are related with the exposure that we have to this hazard, and also the vulnerability itself that we have as a society. And what happened there, if we can see in the part of the what is the feedback effect, that finally, because of this vulnerability, this exposure that I got to talk later, we are going to have climate sensitive disease outcomes, like vector-borne disease, waterborne disease, another kind of infectious disease like a heat-related illness, mental health, and undernutrition. In addition to that, we have migration and displacement and conflict. This is a kind of very heavy burden, a lot of vulnerable people, but also to the government that had to deal with that. But also not just the individual level, it's also at the system that is already impacting this kind of risk. For instance, we have the health system, because we have a lot of patients, the emergency response cannot, serve all the population that it needs, the cost that's increasing. And the pandemic in the 2020 and until now, it shows us that our health systems are totally fragile. So we really need to pay attention to this situation. Food systems, it's not evident, but it's also there. And the livelihood system that we are facing because of these multiple hazards that are going into the same time. But looking also at these climate things, and looking also at the information that we have in the report number 2, the heat risk let's see, with the human health impact, is associated with a range of health issues for urban residents that is unevenly distributed across urban population. But this is very high confidence. So a lot of scientists, we agree in that. And what happened with that is we have high levels of mortality in elderly population from this urban heat and during heat wave event. What happened is because we don't have evidence and data. A lot of people die and the people doesn't know because what? What was the reason that happened there? But also we have vector-borne disease, and climate change affects the epidemiology of climate-sensitive infectious disease in the region. Just to mention that in 2020, Paraguay had more than 200 cases of dengue. So warming temperatures were increasing, and then suitability of transmissions is also spreading for different regions. Finally, the waterborne disease, is there, but it's also expected to compromise because of the WASH service, that is water and sanitation and hygiene in our cities, because it is a compounding vulnerability and increase the water-related health risk. So this situation, we address it using this framework of vulnerability and risk that we use in our research. And the definition that in this moment I try to put it here and display in our case study is that the risk is a combination of the hazard, in this case climate hazard, the exposure of that hazard, and the vulnerability. This is a combination of the social condition that we have. And that vulnerability is made of sensitivity of the population and the system, and the capacity, the adaptive capacity also of the system. Let's see what an example, this case study, we have been working a couple of years at city level to understand the issues of the vulnerability, looking at either climate hazard, like flooding, landslide, urban heat in the city. But to me, it's very important to mention that vulnerability, it's human vulnerability, social vulnerability, city vulnerability is driven by certain sustainable development patterns, such as sustainable ocean or water, land use inequity, marginalization, historical and ongoing patterns of inequity, such as colonialism that we have, and governance. So this is important as a scientist, that we want to make a message to our policymakers that we understand the connection of what are the drivers of the determinant of health in this really broad context. So from there, I'm not going to go into details of my research, but I want to go to the findings of our research. Looking at the vulnerability of this coastal city, this is in Guayaquil, Ecuador, just in the estuary of Guayaquil. Yeah, here, here is Guayaquil, and Duran is just across the Guayaquil, just in the Guayas estuary. And then we are looking at all the city level that has a lot of channels. And finally, we wanted to find where are the most vulnerable population to this hazard, but why they are vulnerable. And one of the reasons, or one of the cause of the vulnerability is this, that probably everybody will expect, because there are informal settlements. There are informal settlements, and we see this map of the city. You see the part that is on a little point, is the area that the city is informal. And it's more than 40% of the population, the area of the city, 44%. So that means that this area doesn't have any service, no water, no sanitation, no road access, so this kind of system. But also we found that the distribution of the demography of the population in a city is very critical. Here we see population over 65 years old, and here we see population under 14 years old. The children and the young family, where are located? Are located just in this informal settlement. Looking at this kind of system, of course, this has put a lot of burden in their health and vulnerability. On the other hand, elder people is in the kind of consolidated city, part of the city, but have, sometimes impacted by another kind of issue. So looking at this, if you remember this combination of safety and adaptive capacity to conform the vulnerability, we found using geographic information systems and analyzing a census tract data with a lot of information and variables to identify where is the population more vulnerable and what are the reasons of the vulnerability. We found, in addition, that informal settlement is the household of quality. It was in the first place, the road access and the lack of basic service. There are kind of the three main reasons that the people are vulnerable, no, in this case at the city level. But also we found this is adaptive capacity that the population has a very limited or no access to health service in this area. So that's the reason that the population has a very high vulnerability in that specific area. And we see in this map, we have like the vulnerability map, and we saw the highest area of vulnerability. And in this map, that is kind of the urban kit, we see the distribution of some illness like IRI, like infectious respiratory disease. Also the green one correspond to vector-borne disease. So we saw that the distribution of the diseases is different in this different part of the city. However, we also find a very lack of information in these areas that are informal settlement because there is no any medical center, no public health center in this area. So we really need to find a more disaggregated information about how is the epidemiology of the people at the city level in order to try to do some specific action to the specific measure. So we see that the vulnerability is in different parts, but also we saw different kind of very social issues related with this vulnerability. So what are the alternatives we are looking? So we are talking about the climate hazard, but also we are looking also for the climate development, how will be, and adaptation and mitigation in the context of the sustainable development is a measure that's coming also from the working group 3, the group that I have been working, in order to try to put as a framework that the society, the city, the decision maker can use it. Because this can be used as a template to evaluate these SGDs. And also we need to understand when I'm looking at that, the co-benefits and trade-off associated with this mitigation, but also with adaptation to try to do the to prioritize what are the actions that we need to do. In this diagram that coming from the working group 3 in the IPCC last report, last month, we saw that, for instance, the urban system is an opportunity to really develop the SGDs. And we see here number 3, that is human health, and mostly all of them are plus, different color. What it means, we can do a synergy. If we are doing land use planning, spatial planning, we will have a synergy with human health, with positive effect. The same with energy, with cooling and heating network. The same with urban green and blue infrastructure. No, the same with the wash, no, the waste prevention, and also this integrated sector. So that means that we have an opportunity. It really, we see in this systemic way, looking at the different systems. And this, we are going a little bit more in detail, like the SDG 2, we're looking at the health and well-being, we will need prevention medicine, identification of vulnerable, surveillance and early warning for this epidemic, diagnosis of emerging disease, and the nutrition and non-communicable disease. We need to know this in order to put effort to prioritize what are the principal action that we need to do. And the SDG 10 that is critical is the investing in universal basic infrastructure. This is kind of the minimal thing that the society have to invest in the cities and in almost every human settlement. That is sanitation, clean drinking water, drainage, land rights, increase the adaptive capacity, because with that we can reduce the vulnerability with climate-related risks. And the sustainable city have to put in the policies and the design to facilitate like a urban active transportation, like it means walking and bicycle, that will reduce emissions, improve air quality, and generate physical and mental health benefit for the residents, reducing the risk for heat stress and respiratory illness. And finally, all the ecosystem that we have in the city, like a land and water ecosystem, we can use it as a nature-based solution for water-related adaptation using urban green and blue space that contributes to adaptation and mitigation at the same time that improve physical and mental health and well-being. This is a very high confidence kind of a statement. So it's important as a researcher, we really can start working in this kind of issue integrated. So the final thought that I have in this case is like the COVID pandemic gave us a good lesson, a big lesson that hopefully we'll learn, have really revealed the limited articulation of the health sector, the disaster risk reduction, the city and the subnational government with the different stakeholders, with the private, with the different, it's limited, it's still this kind of articulation. So it is, in addition, as a scientist, it is necessary to develop integrated data and information from different sectors. So now is the opportunity to establish a strong collaboration between sectors, ensuring that knowledge, science, and data are available to all. Then it's possible to determine action to reduce vulnerability, multi-hazard, and also go to achieve the sustainable development goal. A final measure that is from the Dr. Carissa Chin, director of PAHO, is like without the ability to detect, respond to, and mitigate the impact of the pandemic and other public health emergency, like the vector-borne disease of any epidemic, sustainable development simply cannot be achieved. So thank you, and that's kind of my presentation. Thanks a lot.
Thank you very much, Dr. Mercy Barbour. Very interesting presentation. Now we will continue with our next panelist, who is Dr. Washington Cardenas. He got a PhD in environmental and evolutionary biology at the University of Louisiana Lafayette in US. He has a postdoctoral training in New York University at the Department of Molecular Parasitology in New York, where he was working on malaria vaccines. He also has a postdoctoral training in the Icahn School of Medicine at Mount Sinai Hospital in New York, where he was working on the molecular mechanisms of innate immune evasion by RNA viruses. His research interests are molecular virology, host immune response to viral infections, molecular diagnostics, and vaccine development. Please welcome Dr. Cardenas.
Thank you, Carlos, for the presentation. Let me share my available to share my presentation.
You are as a co-host, so you should be able to share the presentation.
Okay.
Can you?
Okay. Yeah, I think so.
Okay.
Let me think.
Yeah, it's coming.
Okay, perfect.
Yeah.
Okay, well, thank you for the presentation, Carlos. And yes, my main work is basically with RNA viruses, which is a really very important topic in health right now in the world, as you can see. And let me see. And I think in my talk will be in health sovereignty to secure good health and well-being in developing countries. And some Mercy made a good point in her presentation when he finished, was that without having some health sovereignty, what I mean with that is that be able to very precisely to able to diagnose and try to develop probably biologicals to secure our health, and not be in the position like happened in this pandemic to try to wait for all reagents and vaccines or that are being developed everywhere else. So that we won't be able to secure our health in at least in developing countries. That's the main point that I would like to address in here. And also being a developed country like Ecuador is, doesn't mean that it's not capable of a very high standard of technique development in health. So that's, I will hopefully when I finish, you will see that in Ecuador, we should be able to at least for the next pandemic, I hope, be able to be better prepared than this COVID-19. So just as a little snapshot of what the importance of these viruses are in the world in terms of human health. I start with this, let's go up, down, up. We can start with Spanish flu. I guess everybody probably knows about this Spanish flu, and at least this pandemic was good in the sense that a lot of people become very knowledgeable of the science behind and the importance of knowing these viruses. And Spanish flu was the first pandemic of the 20th century that really was a very even more impact the human health than the actual pandemic. Actually, that lasted the same amount of time so far, but that was a lot of human deaths in this Spanish flu that is not being seen in COVID right now. Then we have well, Ebola fortunately has not left the African continent, but it's a very scary disease that all the time you can see outbreaks of Ebola and also with a widespread distribution in the African continent. So that's something we need to take care of because of very high fatality rate. And this is also an RNA virus as well. HIV, well, all we know that we still are in the pandemic, very long pandemic of HIV, which is also an RNA virus. The difference of this RNA virus, of course, is that at some point of the viral replication, they go through a stage of DNA. So that's what makes it different from all these lists. But still, I mean, we can classify this as an RNA virus. SARS, as you know, the first SARS epidemic that was also scared the world because it was probably becoming the new pandemic. Also, the first coronavirus also fortunately just last very short period of time, but it was really a lot of fatality rate. Also, it's an RNA virus. Then we have the flu, the pandemic that we were waiting for a long time. We saw it was going to be avian flu, but then we have the swine flu that we call, because the recombinant virus, also influenza virus in 2009. That's also respiratory virus. That also, again, is an RNA virus. And then we have two arboviral epidemics that also scared the world. There was Zika and chikungunya, both transmitted by mosquitoes. And also that was, at least Zika was really a adaptation of public health because of the problem they have in developing of fetus on pregnant women. And finally, we reach the COVID-19, the pandemic that everyone is waiting for. I mean, at least that was predicted to happen. In COVID-19, fortunately, the world was, I guess, very prepared in terms of trying to identify faster than happened with SARS, for example, what was the etiology agent. So we have, in record time, we have a sequence of the whole genome of these viruses, so that make it possible to right away develop regions for either diagnostics and also to develop vaccines at the same time. So we have to take present that the fast development of the vaccine for COVID-19 was not because people start doing vaccine at the time of the pandemic. Actually, the previous pandemic, Spanish flu, swine flu, and the SARS, people were already developing reagents to try to make platform for rapid vaccine development. But all these platforms didn't go too far because all these epidemic or pandemic just start short. But then with this COVID-19, actually the term around all the platform that was already developed for COVID-19, that's why we have this very fast vaccine that we all have probably taken. So in general, all this, as you can see, again, I want to address again that all these viruses are from RNA in the genome. And just to put in context what happened with Spanish flu, the 2009 H1N1 pandemic and this pandemic right now, all RNA viruses. We have the Spanish flu last about 3 years because we start really early in 1918, so we can take that in all this year. We have to count We have 500 million infections, which is similar, if you can see, all the way down to the coronavirus right now. We have now 513 million infections. But the death rate, of course, is quite different. We estimated that the death due to Spanish flu was around 50 million around the world, which make it so far the most, in 20th century, most devastating pandemic. The pandemic of H1N1 2009, which only lasts about probably 1 year and a few months, will replace the H1N1 that was circulating before, and also it infect at least 200 million, but right now it's everywhere. Like all the probably most of the H1N1 that is circulating in the world is already a pandemic 2009 strain. But the death rate was around from 151,000 to almost half a million deaths. But it's curious that if you see this death rate of H1N1 2009, more or less similar to the seasonal flu that happens every year. There's more or less the same number of people that die every year due to flu. And now finally we have COVID that we are still, started more or less in January 2020, but it was identified, I think, in December 2019, sorry. And but still I put a dash in there because right now still officially it's a pandemic, even though we are already relaxing in terms of some behavior of some use of masks and everything, but still it's I think officially has not been over yet. We have, as I told you, we have about 513 million people infected, similar to the Spanish flu, but as you see, quite different in terms of fatality rate. And this was, the history in Ecuador that hit very hard pandemic, because of course we have a very poor development in terms of technology in diagnostics, and of course antivirals development. You can, even vaccine, we have zero production in the country. So that's why we have a very, very bad situation in Ecuador at the time of the pandemic of 2020. So the first case, of course, in Ecuador was officially detected in February, at the end of February 2020. And the reason why it takes so long to try to set up that's not diagnostic, because it's the first tool that we health can use to more or less follow up the spread of the virus and infectious diseases. We have to wait until, because of a policy, have to wait until it comes for official international health organizations in order to be able to use it in Ecuador. Even though right now a PCR diagnostic considered something that can do this very basic tool for research and for diagnostic. So right now, the accumulated cases of COVID-19 is about 870,000, and we have official numbers are these, and we have a death rate of about 35,000. Of course, these numbers are considered a subestimation because all these numbers are the ones that are being diagnosed, but there's so many more that have not been diagnosed, and then this is considered to be higher than that. But we have good news after all, because so far we have about 80% of people fully vaccinated at this point in Ecuador, and there's still a few people that, of course, doesn't believe in vaccines. That's really amazing in a developing country. But I think we have a very good percentage anyway of vaccines that were given to adults and now children. That's fine. Now, as we know, we start already in the coast region of Ecuador. We start classes, and then we switch to presential classes right now. So which is good, because we know that all this COVID pandemic not only hit in terms of health, but also it was a big problem in terms of psychology of people that were, even worse, children that was really secluded and they need interaction with their peers. So in terms of technology, a few of the basic things that any country in Ecuador, at least Ecuador, it should have, is be able to do their own diagnostics. diagnostic like RT-PCR, which I told you before, is something really basic to do. It's not something that requires a high-tech environment, was missing in Ecuador. So we have to wait again to have the reagents from outside Ecuador to be able to start doing the running diagnostics. Of course, we didn't have serology, which is basically either a point of care test or ELISA test that also we have to import in order to do also follow up the serology of the people that was infected. And finally, of course, worst, we weren't expected to have a vaccine developed in Ecuador at all, because we didn't have basic technology. Worse, we're going to have vaccines. But all these 4 points that I show you here, that Mercy again make a good point. The last presentation show that we require all these, at least all these 3, the first 3, to be able to follow up any disease outbreak and to have at least some kind of health measurement and try to stop the spreading of the virus or any infectious disease. We have in different universities in Ecuador, we have the capability to do that, but yet we are in the position that we need to buy everything outside. So what happened during this pandemic, I think we have 3 universities and 1 institute of health in Ecuador. We took the challenge to try to change this, to make sure that for the next time we at least have a prepared platform, at least a proof of concept, that we don't have really to be waiting for reagents to come to Ecuador. As you know, these reagents were also in high demand everywhere in the world. So even though if we have money to buy and import these reagents, they were already used up someplace else. So there was a big problem also in terms of try to get enough production to import in Ecuador. So, but anyway, so these 3 universities and Institute of Health in Ecuador took the challenge to change this. So what we did is we tried to set up in-house real-time PCR to detect COVID-19. Also, we are right now developing in-house also serology diagnostics. Also, we are working with a university from the US to try to test in-house rapid tests, either to detect antibodies or antigens from coronavirus, for example. And finally, even amazingly, we also took the challenge to try to develop a local vaccine. So all these 4 things that are key to any country to try to defend themselves, confront an infectious disease. We all, amazingly, in 1 or 2 years, we cover all these 4 points in our research. I'm going to show you at least one right now because of the time. So as you know, this is a virus that I guess everybody in the world right now is very expert in coronavirus. And we know that this virus have these famous spike proteins outside, which is basically the key that the virus used to find the lock. The lock should be a protein in the human cell that are required to enter and infect the cell. So the basic concept right now for vaccine development, which is, of course, the more logical thing to do quick, was to try to block this key to enter the cell. So all these, most of the vaccines that probably you already have used are based on the spike protein, which is these little spikes, red, that are outside the virus in here. So this is a big protein, and so you receive either an mRNA vaccine, which basically is the coding sequence of the spike. Eventually, the mRNA will go inside the cell when you have the vaccine. And this mRNA becomes translate to the protein, which is now the spike protein. Probably other people use the vector vaccine, which is the AstraZeneca as an example, which use a vector, which is adenovirus that has inside of the genome engineer also the open reading frame of the spike protein. So the virus is injected in your body, and then it goes inside the cell. And of course, the virus is replication deficient, only will produce, but nevertheless, it will produce the mRNA of the spike, and then we have again the protein spike. The other vaccine that is different is the the ones that have inactivated viral particles, which is actually the actual virus, the coronavirus, which are being inactivated with the old-fashioned vaccine that we used to have a long time ago. And basically, they grow the virus in cell culture, they purify the virus, and they inactivate the virus chemically. So then they inject you the whole viral particle to your body. But again, the main proteins are going to be outside of this inactivated virus are the spike proteins. Okay, so the philosophy behind this is try to make the body makes antibody against these spike proteins, so it will block the interaction of the spike protein with the receptor in the cell. So then you avoid the entrance of the virus. That's the basic concept. So what we did in Ecuador, we went with this group of universities that I will show you later on, who are these people that took the chance to do this. They say, okay, let's do something different, because since the 2009 influenza pandemic, we always stop in the diagnostic part of the problem. Never went further than that. So I'm trying to find a remedy. And the Ecuador had a history of making vaccines previously. So we say, why not we try to do something different and also something that make a long-term solution to the problem. So we try to, again, to the challenge to do this, which was a lot of criticism, and probably, but still we did it. We tried to do it because in Ecuador, as I'm trying to stress, there are different universities that are well equipped with very high standard equipment, and also people that are very well trained in other universities in the world. So there's capacity to do that. So we took this challenge and we took this model, as you see here in front. We took the model of the antibody, human antibody, which you see on the left, which is IgG1. As you can see, let's see if we can show here. Here you have IgG1, you have a little a stalk here, and then you have the streams like a Y layer, where the streams, the end of the streams, which are this orange, couple orange ovals, are the ones that interact with the pathogen. Okay, that's how the antibody works. So the end of the Y, these orange ovals attached to the pathogen, and the stalk, this part in here is the one that signals to the immune system that the antibody has attached to a pathogen. Okay, and see this part is recognized by the receptors that are in immune cells. So the immune cells go by and see that the stalk part of this Y, the antibody, have something happen to it. It has changed the shape because it's attached to a pathogen. So immediately it captured the antibody and the pathogen or antigen that is attached to the antibody. Okay, so the signal is given by this part of the stalk of the Y, this part here, the last part. So what we did is that, okay, we took this antibody apart. We took out the parts that recognize the antigen, but we kept the stalk part, the ones that interact with the receptor in the immune cells. But instead of the antigen binding site, we fused it to the receptor binding domain of the spike protein of the coronavirus, SARS-CoV-2. Okay, so the receptor binding domain is this section of the spike that directly interact with the receptor or the human receptor to get inside the cell. Okay, the spike is a big protein, but all this protein allow the virus to show the receptor binding domain and interact with specifically with the receptor. So if we block this little part, the receptor binding domain, we should be able to block the entrance as well into the cell. So we choose the little part because it's a small part of the protein, it's easier to express, it's easier to clone, and we fuse this to this section of the IgG, which is called the Fc portion. Okay, so because the antibody has two binding sites, so also this expected that this construct, this recombinant protein, will have also two receptor binding domains as shown in here. So of course, you do all this construction in a DNA form, and then you transfect this DNA into a cell. The cell will produce the protein then is purified, and then after that, we go ahead and inject in mice to see if the mice actually can recognize, the immune system of this mice can recognize this recombinant protein and generate antibodies against this protein. So the the concept is that these antibodies that the mice produce should be able to recognize, of course, the receptor binding domain, like would happen in a human, and block the interaction with the receptor AC2, as you know, in the human cell. So that's the model. This is another model that we use. That the only difference between the previous one is that it's have more repetition. As you can see here, this is what is called a CH2 domain. Have several repetitions of this CH2 domain, but at the end we still have the receptor binding domain fused to this CH2 domain of the antibody. Again, you produce this in a cell, purify, inject, and see also if the antibody are produced in the mouse that recognize this receptor binding domain. So the system is really simple, as you can see here, because all the technology is already developed in the world that makes it very easy to work with these proteins and purify them and get it pure enough to be able to inject directly to the mice without too much sophisticated equipment. So basically, what we did is again, we took the plasmid. The yellow part of the plasmid represent the upper reading frame of the recombinant protein that I showed you before. We introduced this in a culture of cells. This is a mammalian cell. And then the cell will, of course, produce the RNA, messenger RNA of the plasmid, which is a recombinant protein. The messenger RNA will be translated into a protein, and the protein will be secreted into the culture media, as you can see here, okay? So on purpose, we make it the cell to secrete this protein because that way make it easier to just take the liquid and purify it. Because this recombinant protein has the Fc portion of the antibody, it will be easier to just get magnetic beads that are covered with protein A. Protein A is a bacterial protein that have high affinity for the Fc portion of the antibody. So if you put in here just in a test tube, it is a microcentrifuge tubes, 1.5 ml. You put these magnetic beads with the liquid, then all these recombinant proteins that are produced will attach to the beads, and then you can wash without centrifugation easily. Wash several times in order to get very pure protein that are attached to the beads. Then after you, of course, separate the beads from the recombinant protein, and then you supposedly have purified protein, and then you go ahead and send it to inject to these mice and check if actually have an immune response. So this will be in collaboration with ESPE, with the university, Army University, which is in Quito, working with Dr. Maribel Torres. She's my partner. She does the preclinical part with the animals, and we just ship this protein to Quito. This is more or less the framework that she used to get a quick check if the animals were actually responding to this protein that we purified, and we design the lab, we produce in the lab, we purify, and we send it to Dr. Maribel Torres in ESPE. She basically did a quick immunization in which, time zero, they just bleed the animal to make sure they have a blood time zero without any injection. Then after 7 days of immunization, she took some blood, and then after 14 days, took again more blood of the animal to see, when the antibodies were, you can pick up the signal of antibody produced because of the antigen that was injected. This is just a preliminary information here. Of course, we already passed this and we have more data, but if you can see here, this is one model, the RBD for CH2. This is the one that I showed you, very, very tall, that repeats several times the CH2. And this is the other model that I showed you, the small one, which RBD with the FC, with the basic antibody FC portion. If you see on the left, you can see more, you can see already, we can see a signal at, this is, I think it's 7 days. After 7 days, and then we have 14 days. At 7 days, we can see, you can see here on the left, this orange circle and the purple square, we can see already a signal at 7 days using the RBD for CH2, which means that the big model that we use. But also at 14 days, we see also a signal. This is only positive and negative. This does not quantify the antibody. You saw this positive and negative. All these little squares and triangles you see on below are basically background to show you that this is a real signal. We use several measurements to see if, what's the background that we may have, on the non-specific signal, what it means. And you see on the right, the other model, which is a RBDFC, the small one, it was not as striking as the big model. You can see a signal, but you have a large standard deviation, a lot of noise of using this RBDFC. Okay. So anyway, but this was really exciting to us because actually we produced a recombinant protein in the lab that was immunogenic and that was detectable in these mice. But the big question was, okay, are these protein that we're producing in the lab, which is recombinant, which is the receptor binding domain, of course, it's not the whole spike. And then we are expressing this fused to another protein, which is the Fc portion of antibody. The question is, are these still protein being expressed in a way that can be still recognized, but antibodies that are developing people that actually had an infection? That will make a good point that yes, this protein is relevant. This recombinant protein is relevant because it's being recognized by antibodies of people infected with coronavirus. So that's what we did. So we did an experiment in which we took a little, this is a filter paper, which is very usually allowed for doing immunodiagnostics. And we print in one of these, the model that I showed you before, our receptor binding domain fused to the Fc portion of the IgG1. Then we fuse next to it, we print the other model, which is the one that have several repetitions of CH2 domain, and then still we have the receptor binding domain. We put then a irrelevant protein here. This is a extract we did with the same cells you saw before, the human cell, we expressing this antibody, but we transfecting the cells the green fluorescent protein, and we purify that, and then that's a irrelevant protein, of course. And then as a positive control in here, we put purified IgG as a positive control. So now to see if then you took the sample of serum sample of a person that was positive for COVID-19. So if this person had, of course, was developing antibody against the virus, it should be able to hopefully recognize and attach to the receptor binding domain that we have in here. So this is the spectral result. So these purple antibodies represent the person that already was infected with coronavirus and supposed to have antibodies against, of course, the spike protein, also to the receptor binding domain of the spike protein. It should attach to the model 1, to the model 2, of course, but not to the control and not to the IgG, of course, purified, because it's not coronavirus antigen in there. Now, in order to detect the presence of this human antibody that has attached to our receptor binding domain, we should use a signal. This signal is given by another antibody, commercial. You can buy a commercial antibody that is recognized human antibodies, okay? So if there is any human antibody attached to our printed recombinant protein, this antibody have the little signal should get a capture in there, as you can see here. Of course, it should happen also in the M2, in the other model. It shouldn't happen in the control, but it should happen in the IgG purified, because this is a human antibody that we put on purpose in there. Purified human antibodies should attach this antibody with a signal because it recognizes human antibodies. So now this is the spectral result. That what should we, if everything goes okay, it should have signal in N1, we should have signal M2, we shouldn't have signal in C, we should have signal in IgG. And that's the belt we use, of course, chemiluminescence kit to develop this, and the result is as expected. We can see signal in M1, we can see signal M2, we don't see signal in the control, and we, of course, see signal in IgG, the purified IgG, which means that that's very exciting, because that shows that our recombinant protein is being recognized by serum of people that were infected with coronavirus. So of course, we did this also with serum with people infected with, we have in the lab with dengue, where also people, I talk about blood sample from probably 3-4 years ago or more, that of course that was not this virus circulating at that time. And that was clear. It was there was no binding of those serum with M1 or M2. So that's what's really exciting to us, because it shows that these antibodies are recognizing this receptor band domain. What is interesting, the reason why we did this essay is because it seems that when we do the Western blot, I don't know if you're familiar with Western blot, that you go through SDS page, which is you denature the protein in order to do the Western blot to make this assay, you cannot see any signal, which means that these epitopes, that these receptor body domain are conformational, that requires to be innate in somehow in not denatured in order to be recognized by the human antibodies. So just I'm going to stop right here. We did all this work in collaboration with ESPE, UTLA, and INSPE. In ESPOL, we have very smart students and great technicians that were working on this. That you see the names: Mariuxi, Maria Jose, Cristina, Michelle, Fernanda. In ESPE, we have Marbel Torres, which is the lead scientist in there that is working with the biopsy mouse to do the preclinical assay that we're still doing right now. Esteban Ortiz is very good. It's a medical doctor, which is very good in terms of clinical trial. Hopefully, we can go all the way to clinical trials someday. And of course, INSPE is a key collaborator in here, because actually this is the Research Institute of Ministry of Health in Ecuador. And we have a collaboration there with Leandro Patiño, which we work together in this, as you told before, a challenge. We got funding from CEDIA, which is very good. That was the only agency that be able to give us some funding so far. They really believe in our challenge. So this is all I have. If you have any question, I would be happy to answer after we finish.
Thank you very much, Dr. Cardenas. I believe we learned a lot from your presentation. Next, we are pleased to introduce our final panelist, our special guest from Florida Atlantic University, Dr. Krista McCoy. She received her Bachelor of Science from the Old Dominion University and a Master of Science from James Madison University, and her PhD from the University of Florida. She has a broad training in ecological development, toxicology, and behavior. As a postdoctoral scholar, she was awarded with an NRS fellowship from NIH, and then in 2013, she joined the faculty at East Carolina University. She had an active NIH funding research program there, and mentored several graduate and undergraduate students in research. She was tenured and was a founding member and president of the Eastern North Carolina chapter of Graduate Women in Science. She joined the faculty of Harbor Branch Oceanographic Institute in the summer of 2021 as an associate research professor, and she is currently funded through the HBOI Foundation. She is also an active and funded member of the Florida Center for Coastal and Human Health, and her current research focuses on understanding how to prevent common disorders induced by exposure to environmental pollutants. Please welcome to Dr. Krista McCoy.
Hi, thank you so much for inviting me. I'm sorry that I don't speak Spanish, and it's my worst character trait. Let's see. Okay, I just want to make sure that everyone can hear me. And so if someone can just let me know, I will get...
Everything okay. Looks fine, and we hear you clearly.
Great. So I love the last talk, because one of the things that unites our talks is this idea of coming up with a different perspective or a different way to solve a problem. And so today I want to talk to you about developing a new perspective in the study of ecotoxicology. And I believe that that new perspective will help advance health and well-being. oops, let me see. Okay, so we have about 70,000 synthetic chemicals in use on Earth currently. And only 2% of those have been evaluated for human health effects. And about half of those that have been evaluated are known to disrupt the endocrine system. And these endocrine disrupting chemicals are pervasive. They are in our drinking water and in our wastewater because of medicines that we take. They're intentionally sprayed on our food as pesticides. They're impregnated into materials that we wrap ourselves in. They are also in plastics that we give to our babies to suck on or to eat from. There's so many of these plastics that they're literally forming mountains. And these chemicals are also in the air. And they're also in our bodies, not surprisingly. And so this little figure shows the percent of Canadians tested who had flame retardants in their bodies. And so this little person is shaded to show the proportion of people that had this particular PCB in their body. And in this case, it was 98.6% of the people tested. This PCB was found in 100% of the people. And so these chemicals are all over and they're inside of us. And we also intentionally spray many of these chemicals on our food. And those are the chemicals that I worked on during my dissertation work, and these endocrine disrupting chemicals that we ingest through our food. And the endocrine system turns out to be extremely important for development. And we know that embryonic exposure to endocrine disrupting chemicals correlate with the incidence of birth defects in humans. And one common birth defect is called hypospadias, and it is a birth defect of the penis. We don't talk about it a lot, but it's actually very common. And so this happens when the urethra doesn't open at the distal tip of the penis, but rather along the shaft or even in the perineum. And this birth defect has increased 500% over the last 50 years. And it affects nearly 1% of boys that are born. And in some cases, it's almost 5%. And so we know that this birth defect is associated with embryonic exposure to pollutants and endocrine disrupting chemicals in particular. So I began studying how endocrine disrupting chemicals affect genitalia development. So how are these chemicals causing those developmental abnormalities? And I used the mouse as a model to try to learn how humans might be affected. And we've studied this over the last decade and I think have made some interesting discoveries. The problem, though, is that knowing the mechanisms through which chemicals induce harm doesn't stop that harm. And so what I decided, and my lab has decided that we need to kind of change the question. We need to stop only asking what mechanisms induce toxicity, and we need to start asking, how do healthy phenotypes develop in polluted embryonic environments? So how do we develop a healthy baby even if our bodies are polluted? And so one way to think about this is just the sort of theoretical dose response curve, which are common in toxicology. So we have, this increasing dose of some contaminant, some toxicant, and then we measure the response. And in my field, what we focus on is the mean. And many times we look at that variation around the mean and we consider it nuisance variation. Something went wrong. or, there's just variation in the way that organisms respond. And we don't really think about why that variation exists. And I became interested in these individuals, focusing on the individuals that responded the least to, the chemicals that we were studying. And my question was, why are they resilient? Maybe they have antioxidant enzymes or detoxification enzymes that are expressed at a higher level than the other animals. And then that led to this idea that if we can increase those enzymes in our bodies, maybe we can increase resilience to the chemicals and decrease the response to them. And so I have this MC Escher picture here just to emphasize that I'm arguing that we need to look at the world in a different way, look at this field with a different perspective, and begin to study the mechanisms of resilience. And to begin this, we decided to test whether we could develop a prenatal supplement that would reduce the effects of toxicants and protect the developing baby from these toxic chemicals. And we've worked on this for many years, and we've developed a model system where we can use vinclozolin, which is a pesticide that's sprayed on a lot of different types of food. And we can use high concentrations, and we can cause hypospadias, the birth defect that I referred to earlier, 100% of the time. And that allows us to have a very strong model for testing whether certain supplements can reduce the number of cases of hypospadias. If we know 100% of the animals should develop it, then any treatments that reduce that number are interesting. We also standardized a scoring system so that we could quantify the severity of hypospadias in our mouse model, so that we could compare different supplements, and also so that different labs around the world could use a single scoring system for mouse studies. And then after conducting several different response curves with different supplements, we settled on focusing on sulforaphane, which is a chemical that our bodies produce when we eat broccoli or other cruciferous vegetables. And so remember, we're trying to ask, if we give this sulforaphane supplement, can we reduce the response to environmental chemicals such as vinclozolin? And what we found is that sulforaphane can reduce this dose response curve to vinclozolin. Without sulforaphane, we get this dose response curve that is typical in our lab, where high concentrations of vinclozolin cause 100% or an probability of 1 of developing hypospadias. When we give sulforaphane, we can reduce that dose response. And when we look at severity of those individuals that have hypospadias, we can reduce severity as well. And so that's really exciting. And now what we're doing is investigating the molecular mechanisms of resilience. How are those individuals able to develop less severe hypospadias and less cases of hypospadias? What are the mechanisms through which sulforaphane is working? And I don't have time to talk about that today. But what I would like to do is talk more generally about this idea of developing mechanisms of resilience and thinking about how we can more directly protect ourselves from environmental hazards. And so we know in the case of sulforaphane that it does work in humans. We have, there is a lot of data on sulforaphane, and in many different contexts. And it's sold now on Amazon, that you can buy sulforaphane supplements. And so it's safe. It's used in over 70 clinical trials right now, looking at its effectiveness. But what is not being studied directly is its ability to be generally protective for environmental contaminants. And so we've shown in the mouse that it can protect against vinclozolin, but really the next question is it generally protective? And so what we are asking now is whether sulforaphane can protect against toxicant-induced autism. And so we have NIH funding to answer whether or to try to understand the neuroprotective effects of sulforaphane in VPA-induced models of autism. So VPA is valproic acid, and this is a chemical that is given to people that have epilepsy. And years ago, when women that had epilepsy continued taking valproic acid during pregnancy, their babies were more likely to develop autism. And so people noticed that and then began working in the rodent models to ask if VPA could induce autism-like behavior in rodents. And sure enough, it does. And so we're using this model to ask whether or not we can use sulforaphane during development like a prenatal supplement to stop that autism from developing. And we're doing this in two different ways. My colleague, Karen Litwa from East Carolina University is coming at this problem using human brain spheroids. And she is asking about neural circuit development in the presence of valproic acid or valproic acid plus sulforaphane. The problem with that system is that there's no behavioral readout. And so I'm working in the mouse to do very similar exposures to valproic acid alone or valproic acid with sulforaphane to ask if we can prevent the valproic acid induced autism-like behavior in the mouse. And this work, with this work, we hope to identify conserved sulforaphane-induced genetic and cellular mechanisms that protect the brain development and as well as behavior from valproic acid. And another way that we're testing whether sulforaphane is generally protective is by studying the toxins associated with harmful algal blooms. So at Harbor Branch Oceanographic Institute, where I currently work, we are interested in the linkages between ocean health and human health. And so my lab is investigating whether and how we can protect ourselves from harmful algal bloom toxins. I'm currently funded through the HBOI Foundation to answer the question, how does nutritional status affect the response to blue-green algae toxins? And so we know that diet affects our response to toxicants. High-fat diets are associated with more effects. The microsystems that are associated with these blue-green algae affect, in the mouse model, affect mice more if they're fed a high-fat diet. We also know that sulforaphane can reduce the effects of microcysteines. And so our work is asking if we can use sulforaphane to protect against the health problems induced by a high-fat diet, and also if it protects against microcysteines, and then if it protects against that interaction. And with this work, we hope to identify the sulforaphane-induced cellular mechanisms of resilience that protect us from microsystems. And together, all of this work hopefully will help us identify these conserved mechanisms of resilience that can protect us from many different types of chemicals. So I would argue that we need to develop new perspectives in ecotoxicology if we hope to advance health and well-being. And rather than focusing on how chemicals negatively affect us, we need to also determine the mechanisms of resilience that protect us. And my lab is doing this in a variety of contexts. And I would love to chat more with you about these projects in the future. I do want to acknowledge the very many people that have helped over the years, undergraduates and graduate students, and my colleagues, and also my funding agencies. And as with the other speakers, I'm happy to ask questions or answer questions if time allows.
Thank you very much, Dr. McCoy. We are really grateful for your great contribution to this meeting. And now we will move to our question and answer space, where anybody from the public can formulate a question to any of our panelists. If somebody wants to formulate a question over the microphone, please raise the hand and we will allow you to submit your question. Because this is a special meeting between researchers from ESPOL and from Florida Atlantic University, we also welcome questions from one of our panelists to any other of the panelists that have been invited today. I have a question for Washington Cardenas. Okay, Mercy. Go ahead, Mercy.
Thank you very much for all the presentations. And I really like all this kind of the resilience perspective that Christopher had presented. So talking about the ocean and the human health, and I see that you are working in the blue-green algae. I wonder is also are working in some of the algae that are in the ocean, more like in coastal. We also have the blue-green algae, but we don't have a lot of, I think there is some research, I'm not familiar with that, but I have been doing a preliminary analysis, most of the oceanographic variables related with some of the harmful algae blooms on the coastal area. So I wonder, in that case, how are you working in the resilient and with the population in the coastal areas? I know that some of them have been affected. So what is your approach? Is the same, or how you are doing that?
Yeah, that's a great question. It's so here in Florida, the blue-green algae bloom is really highly focused in Lake Okeechobee. And because of the ditching and the canals that connect Okeechobee to the coast, what can happen is when they move water from the lake to control its levels, they can move the blue-green algae out into the lagoon area. And so that's kind of the connection with the coast. I think that we now, because this happened a few years ago, we are now aware that we need to be very careful moving that fresh water into the saltwater systems when those blue-green algae got stressed and died in the salt water. A lot of those toxins were released. And so my lab is sort of have our feet in both areas. Right now, what we're trying to do is establish exposure. And so that these that the harmful algal blooms are very dynamic. And so they're hard to study, because how do you ask for money and say, I promise this is going to happen, but we need the money now so that when it does happen, we can study it. And so we're trying to do it sort of in a layered fashion. So the first thing we're doing is trying to understand how people are exposed. And so we're working with the people around Okeechobee to study the blue-green algae toxins. And then we're trying to work with people along the Indian River Lagoon to try to understand the saltwater related harmful algal bloom toxins. So we hope to, right now we're collecting water samples throughout the year to try to find out when people might be exposed. And then we're going to hopefully get exposure data from having people wear bracelets that will measure air pollutants, as well as get water samples from people's houses. And then finally, eventually, I would like to measure body burdens. Does that answer?
Yeah, I know it's a very complex thing. So, and it's really, you know, you never know when it's showing and you have to be ready. Yes. Thank you, Krista.
Thanks. Please, if anybody wants to submit a question from one of our panelists, you can either use the chat in Zoom, or you can raise your hand and we will allow you to formulate the question using the microphone. I have a question for Washington Cardenas. Are you there, Watson?
Yes, I'm here.
Yeah. Okay. We learned that in your lab, and with the help of other universities in Ecuador, we are trying to develop a vaccine for COVID-19 in Ecuador. But we also know that in Ecuador, we have had a quite successful vaccination plan for COVID-19 using international vaccines. And we also know that internationally, we have several offers of vaccines for COVID-19. So my question is, if you can explain us why it is important to Ecuador, even though we have several offers of international vaccines, and even though in Ecuador we have a successful vaccination plan, why it's still important for us to continue with working on this vaccine program in Ecuador?
Well, I guess the answer will be for the same reason that other countries have done that before. So that's why they are selling us the vaccine. So the point of this is that, for sure, probably very unlikely that we're gonna use this for COVID. Of course, we see that probably pandemic is winding down. But the way that we design this is that it's going to be a platform. We can just change the antigen to whatever other pathogen we would like to immunize to. So this is the reason why it's so versatile. We did this in that purpose, so we can change the antigen again, and we already have the platform. We can do all the assays with COVID, and will be the similar assay with another antigen. But because of, as you can see, all the presentation, the world is present as a very I guess that picture in the future that we're going to have more often epidemics or pandemics of either a bacterial, parasite, or viral infection disease. It's not going to go away. So we have to have a preparation, otherwise we're going to have the same situation in the future that we have with COVID-19.
Okay, thank you very much for your answer. I don't know if somebody else has a question for one.
I put a question in the chat.
Let me see. Or go ahead with your question.
Well, my question was really, Krista, it was interesting about the autism research. So I'm wondering if this BPA, I think, what's BPA-induced autism? This is the pathway that it causes autism is the same, but I guess all the autism that is going on in the world is a similar pathway, or it's different pathway. So in order to be able to apply what you find with this BPA induced to the current pandemic or epidemic of autism.
That's a really good question. So the mechanism through which we think valproic acid induces autism is oxidative stress by changing the oxidative stress in the developing brain, it alters brain development. That's what we think, but it's complex. And surely not all cases of autism are associated with that particular mechanism. One of the things that we know is that organophosphate pesticides are associated, the data is pretty strong. They are associated with autism as well. So moms that are exposed to these kind of pesticides have a higher chance of having babies that develop autism. And so I think the next step is to look in that system and to ask whether sulforaphane can reduce those autism cases. And because that would be yet another, way to test the generality of sulforaphane, but it's also a way that is more realistic right now, because pregnant women aren't taking valproic acid anymore. And so we've cured that problem, because that was an easy, it was terrible for the people before we realized it, but it was an easy fix because it was one chemical that was causing the effect. The problem is that we're not exposed to single chemicals, we're exposed to mixtures. And so if you eat certain things during the beginning of pregnancy, your baby can develop certain problems. Whereas if you eat those same things later, different things are developing. So different problems will happen or nothing. And so it's really complex. And I think my approach is just to say, we know that environmental contaminant exposure during development is associated with some cases of autism. Can we protect our babies from those cases? We're not going to protect them from genetically controlled autisms. And surely this isn't going to protect us from every single chemical, but it's an approach that people aren't taking yet. Everybody, I don't see very much literature on autism focused on prevention. What I see is, how do we treat the children? How can we diagnose it earlier so that we can intervene once the kids are born? But yeah, and so I think like you, just kind of flipping everything on its head and thinking about it from a different perspective might actually be more helpful.
All right, great, thank you. See, yes, that's a big deal, to try to either find what's the reason of the autism. I think it's not very clear. So many theories and so many things that are related to this. But do you think that the same thing will be applied to Asperger syndromes, or they're probably, it's not, it might be also useful for also Asperger?
I don't know, that's really interesting. I do think if we can control high levels of oxidative stress that are not natural, that are above our homeostatic set point, we will have healthier pregnancies and healthier babies in general. I need to think about Asperger and more. I think that's an interesting question.
All right.
There was just a paper that was published maybe 2 months ago on pregnancy-induced high blood pressure, and that sulforaphane reduced high blood pressure during pregnancy. And so it is, I'm really excited about that. That means that we're moving toward developing sulforaphane as a prenatal supplement. And so that showed that at least, a small dose for a few days was safe during pregnancy. So yeah, I'll think about that Asperger syndrome more. Thank you.
Thank you.
I believe we have a question in the chat. Yeah, there is a question for Dr. Krista McCoy. She is an aquaculture student, and she finds very interesting your study between the ocean health and the human health. And her question is, would it be possible for these algae toxins to be transferred to people through the animals that consume them, especially if these algae are non-toxic to these animals?
We can eat these toxins through our food. Yes, absolutely.
So that's quite possible.
That's one of the mechanisms through which humans are exposed, especially with blue-green algae toxins. The toxin doesn't really cross our skin very, very well, but if we drink the water, if we're swimming and the water gets in our mouths, or if we eat food, fish or mussels, mollusks that have the toxin in them, we can definitely get it into our bodies that way. And pets that go swimming in these areas that are having algal blooms can die because they go swimming. They don't necessarily drink the water, but when they get out, they lick themselves and they get the algae into their bodies that way.
Thank you very much. Does anybody else has a question for any of our panelists? No? Okay, so I believe that with this, we conclude our Sembrando Ideas today. On behalf of ESPOL, I want to thank each of our three panelists, especially to Dr. McCoy for her valuable contribution today. Also, I want to thank all of our attendees, and we invite you to our next Sembrando Ideas, where we will talk about SDG number 4. So have a nice weekend, everybody. Thank you very much.